Welcome, everyone. Good morning or good afternoon to the 3Rs Collaborative Translational Digital Biomarkers Initiative 2026 webinar series, where we're excited to have 3 of our member companies present on the application of their TDB systems in neuroscience research. So to kick things off, I'll just do a quick introduction to the 3Rs Collaborative and the TDB initiative. And to introduce myself, I'm Elisa Stevens, the program manager here at the 3Rs Collaborative. I'm going to start off with a disclaimer that I'm just going to read straight from the slide. This webinar series is made up of companies who may be competitors or customers of one another. Accordingly, nothing discussed during these webinars are intended to result in an agreement on price, exclude suppliers from any market, or otherwise restrain competition. Those participating in this webinar series are instructed to avoid discussion of competitively sensitive subjects, including costs, prices, sales, product marketing, and other confidential information. In addition, the 3RC will not use contact information received during this collaboration for marketing purposes or engage in marketing or sales conduct during collaboration activities. Okay, so the 3Rs Collaborative is a US-based nonprofit whose mission is to advance better science for both people and animals, and we do this through facilitating collaborative 3Rs initiatives with key stakeholders. Our efforts span across the 3Rs with initiatives on artificial intelligence, microphysiological systems, as well as refinement and others. For each initiative, we provide thought leadership, we understand current use and barriers through surveys, and education and practical resources through manuscripts, presentations, resource hubs, etc. So our Translational Digital Biomarkers initiatives collaborates to increase adoption and regulatory use of TDB that refine and reduce animals in research. And TDB are aligned with the 3Rs, particularly through refinement and reduction, where the non-invasive manner causes less stress, and continuous monitoring can also identify faster disease progression, and then through reduction by supporting detailed and accurate data collection and allowing for powerful within subject analysis, allowing you to use fewer animals in your experiments. So here are our current member companies, and we bring together end users, tech providers, and regulatory agents. The initiative has published 2 review papers now on TDB that outline value propositions, strategies for implementation, and a validation framework. And you can see both of those through the QR code or through our website. On our website, we also have the TDB Tech Hub that connects end users with relevant commercial tech providers. We currently have 8 TDB companies on there, and you can filter by the specific environment, readout, species, or scalability that you need. You can also access that through our QR code. And finally, if you missed those QR codes or just want to check out more information on our TDB initiative, you can go to our website and the TDB portal, where you can find our Tech Hub, initiative FAQ and how to join, as well as past presentations and webinars. And then finally, during the session, I kindly ask you to use the Q&A feature to ask your questions. And at the end, we'll have a roundtable Q&A with all the presenters, where they will be able to answer your questions. So if you want to learn more, you can visit our website, or if you have specific questions or are interested in joining our initiative or becoming one of our sponsors, my email is on the screen. Please feel free to reach out at any time.
Thank you again, and good morning or afternoon. Actually, it's good morning for me in California, very early, but well caffeinated for this presentation. My name is Jose Gadea, and I work at UID, Unified Information Devices, VP of Sales and Marketing. And today I'm going to be talking to you about making a case for the use of temperature as a biomarker for various neuroscience applications. So to start with, the temperature actually really plays a big role in different neuroscience applications. In cases of circadian rhythms and sleep, we know that temperature dips at night to facilitate sleep and rises during the day to make us active. And any disruptions in these circadian cycles actually can signal sleep disorders and any aging issues. In terms of cognitive function, lower stable temperatures and greater than the day and night fluctuations, meaning the amplitude, are associated with better cognition. and higher, less variable temperatures can indicate cognitive impairment. Various neuroscience diseases also, when circadian rhythms variate, can be an early symptom for conditions like Alzheimer's and Parkinson's disease. So any disruptions of this can be linked to disease processes and help characterize these processes in research. Temperatures also affect neuronal firing, with small temperature changes, even less than 1 degree Celsius, can influence excitability, and warmer temperatures can often silence neurons as well. And cooler temperatures increase excitability. Higher temperatures also, even within the physiological states, they can be linked to reduced brain function volume and accelerate atrophy as well. So, and we all know that extreme core body temperatures, meaning heat stroke, can cause brain damage as well. So temperature can be very valuable for incorporating into an array of digital biomarkers to look at various neuroscience diseases and biological functions. I'm going to provide several examples of in research and how our technology is being used as well. And I'm going to start with the role of temperature in longevity research. This is basically stemming from a paper that came out of Aging and Disease, where the authors recommend a standardized protocol for incorporating into mouse longevity studies. The rationale is really that they argued that no company incorporates long-term lifespan studies in their human clinical trials, and there could be a gap in this, and companies could de-risk their drug development if they were to incorporate these protocols. So the lack of standardized protocols, could be due to various factors, but there are ways to incorporate them early into clinical development to maximize the value of drugs, and even slow down the biological aging, and discover treatments for these chronic conditions, but also ways to improve lifespan. One of their timelines that they provide in their study is that they can incorporate this early lifespan studies very early into clinical development, into clinical testing, and then continue throughout their pathways. So the study itself, the protocol itself recommends the use of about 250 mice that are heterogeneous, to also incorporate male and females, so that they can discriminate any biases by sexes. And also the treatment could begin early in life or later in life of these animals, in this case mice, depending on the cost and study goals, right? But what's interesting is that their recommended protocol includes body weight and temperature as the recommended as a minimal inclusion into these studies, since they're non-invasive indicators of health, frailty, and human endpoints. So this would be the core elements that would be included, including survival curves, temperature, and body weight. And there could be enhanced options that can also be included, including digital phenotyping, video, AI monitoring with video, taking regular blood samples for measuring different organ function and things like that. So as well as other behavioral technologies to further evaluate the disease. But however, the body temperature and weight are among their core, and they could be very cost effective and now with technologies easy to monitor throughout the course of the study. Also in psychedelic research with regards to depression and anxiety and schizophrenia, there's a study that came out recently out of Monash University looking at psilocybin psychedelic that modulates social behavior in male and female. So they studied both sexes, and they found that there were sex-dependent and time-dependent influences of psilocybin in this study. Again, the main reason for performing the study was to look at the sex-specific differences and looking also at the involvement of serotonin receptor antagonist in modulating the social behavior. So they used male and female mice. They were pair housed and dosed with a single dose of psilocybin or the saline. And they also, in some experiments, they administer serotonin receptor antagonists 30 minutes before psilocybin to see their effect and how it would influence the effects of psilocybin. And core body temperature and locomotor activity were recorded using the UID mouse matrix system and temperature probes. And social behaviors were actually evaluated using the ethovision by Noldus. So I'm going to show you some of the results here. It might be a little bit fussy to see, and I apologize about that. But what I'd like to really point you to is how administration of psilocybin increased huddling behavior in this. This is in female mice. And you can see how it also included those, increased those behaviors, peaking at 25, 30, 35, and 45 minutes post-minute. This is indicated by the significant difference, right, by the asterisk here. So we have here psilocybin administration versus saline. And when they look at the temperature, they noticed that temperature was dropped right after psilocybin administration, and it was maintained throughout about well, it was statistically significant up until this period, but also it was maintained up to 2 hours post administration. And the images right here, the graphs right here indicate how serotonin receptor antagonists, basically 5-HT1R and 5-HT2, they play a role in the huddling. So with regards to saline controls, the huddling behavior was also impacted. And when you administered psilocybin, it also lowered the huddling behavior. And they also looked at their temperature, and those are the graphs here. Again, it's kind of difficult to see here, but you can in this graph over here, it shows that psilocybin treated mice antagonism also lowered the body temperature, particularly at time 0, time 5 minutes, 25, and 65 and 70, where the asterisks are showing. So in terms of the psilocybin treated mice as well, there was reduced core body temperature at 75 minutes, and the 5-H2 receptor increased temperature at 0 minutes and 85 minutes compared to the psilocybin treated mice. So I know that this is a little bit difficult to see, but I'd be happy to share the study with you after the talk. This is in males. So particularly the difference was that in males, the total huddling behavior was not very significant. It was not significantly affected right here. So you can see that there's not a lot after psilocybin administration, and temperature was not also impacted as well. So that was kind of striking to see the differences that psilocybin induces in males and females differently. And also with regards to the control mice in terms of 5-H2 administration of antagonism, it did not significantly affect also the huddling behavior. So that's kind of what was striking about this study. So again, I'm not going to dive too deep into the results, but also summarize where psilocybin, the differences in female and male mice, and looking deeper into further analyzing and characterizing the model, and now associating it to other behaviors that were evaluated with the ethovision. The results indicate that psilocybin enhances sociability and modulates novelty-seeking behavior in females, and that was done through the 5-HT2 and serotonin receptors antagonists, right? And you can see that sociability scores were increased compared to the control when psilocybin was administered, and also grooming behaviors were also increased, as well as rearing behaviors. So in general, social behaviors were increased in females. That was lacking in males. It was different in the sense that it didn't affect any sociability traits or any sociability behaviors, but it did reduce grooming and also rearing behavior in these mice. So really quickly, sorry about that. So just in summary, what they found is that psilocybin, produced distinct effects that were sex dependent in social behavior, and males and females behave differently. The effects were mediated by the 5-HT1 and then 5-HT2 receptor antagonists, or the serotonin receptors, and it shows how researchers should really be using male and females in their studies, and this has been also shown in different diseases and different studies as well. Another example where temperature, as we know, plays a big role is in circadian cycles, circadian rhythms, and in this case, particularly with neurodevelopmental disorders. So the study out of Baylor College of Medicine looked at the maturation of circadian rhythms in core body temperature over a long period of time. So the idea for the study was really to investigate the use of temperature microchips and the UID mouse matrix for monitoring these circadian cycles over a long period of time, right? So, and that's one of the advantages of the system, that it lends itself for long terms of longitudinal evaluations. So they started at peri-weaning, at postnatal day 20, into adulthood of about 75 days. And the idea was to generate an objective biomarker that then could be used for other neurodevelopmental disorder models. So they used wild-type mice and implanted the UID temperature microchips and group housed the animals in the mouse matrix for that period of time. And circadian properties were quantified using the Casinor analysis to estimate the amplitude and the measure of the circadian cycles. And the other parameters, the other matrix, like in interdaily variability, stability, interdaily variability, and other factors were evaluated using these formulas over here. So first of all, I'll show here a heat map that shows the daily temperatures of males and females. This is starting from the day 20 followed up to day 75, basically along the vertical axis. And you can see how male and female temperatures are very different, with the female temperatures being higher throughout the entire evaluation period. And also you can see that there's higher temperatures during the dark cycle compared to the light cycle. And then when they looked at the amplitude and all the other circadian measures, they found that there were gradual increases in circadian amplitude for both, actually, male and females. And there were gradual reductions of the measure, but that only applied to the male mice, not the female mice. There were also gradual reductions in the fractal complexity in females, but not in males. And also there were no other changes in their intradaily variability and also in interdaily stability. So in conclusion, first of all, they found that their results were consistent with other evidence that compared to human subjects, that temperature are higher in female subjects. But particularly what they wanted to do is they look at their patterns of the circadian cycles, how they apply to aging mice, so that they could incorporate it with other models that include seizures, particularly with epilepsy. So they're going to be using this data for further characterizing various neurodevelopmental models that they're studying. And as a side note, we also have investigators using the system for traumatic brain injury models and also Parkinson's disease models. So this data can be also very useful for and beneficial for that. So, I talked about the science. I just wanted to show you how this is capable, right? So there's a lot of high resolution temperature data that is generated, and you can see that there were like the Cousineau analysis shows that there's statistical programs that sometimes I use to really manipulate the amounts of data, but it provides high resolution. So how is this possible? One of the reasons is the probe. So the temperature microchip is minimally invasive, so it's injected with a needle, so it doesn't require any surgery. So this actually improves animal welfare. It eliminates invasive procedures and handling. It lends itself for long-term monitoring, because one single implantation, it doesn't have any batteries, so it can stay in the animal for the entire lifetime of the animal. So you can follow these animals over a very, very long period of time without additional manipulation. It's also very cost effective, and is primarily it's very accurate. So it has the similar accuracy as telemetry implants or rectal probes, which are technologies that are widely used. 0.1 degrees Celsius for very reliable data. And you can see how in the data that I presented, it's very important to really have a very accurate tool, since in some cases very small changes are occurring in some of these studies or some of these neurodegenerative diseases, to be able to detect it accurately. And in terms of, once the microchip is in the animal, so how is this evaluated? We have different choices, different technologies that accommodate for, various studies. So starting with just a simple scan, so to get spot measurements. So this gives you just a single measurement, and as quick as you can scan a button, but this requires in some cases manipulating the animals. And then we have other technologies that allow for continuous monitoring without disturbing the animals. So again, it accommodates for small scale studies up to large scale studies. And with the UAD mouse matrix, which is the system that was used in the studies that I shared with you, it's the one that provides high resolution, continuous monitoring over a long period of time. It's highly scalable. It's adaptable to different technologies. So it allows for really collection of the data that with the high resolution that is needed for characterizing these models. And also it's very applicable to other technologies and can also be integrated with all other technologies, which is important for different research applications, and particularly for expanding the array of biomarkers that can be tracked in a study, right? So this is a perfect example I wanted to share with you, a collaboration, a partnership that we have with TSE Systems with their Promethean core, where they incorporated our temperature monitoring technology directly into their their promethium core metabolic cages. And I'll show you here some data that was generated by TSE Systems, where they're able to very nicely correlate temperature, which is shown in red, to their energy expenditure. So vary that as running speed or running activity is being induced here. So very nicely correlating with that. So again, this shows a very nice partnership. And at the end, really, it's about the science. It's about meeting customer demands and facilitating research and expanding, improving animal welfare. And that's what we're all about at the 3Rs, enhancing animal welfare by combining technologies and reducing the number of animals and so on. So with that, I just wanted to conclude and summarize that temperature plays we know that we discussed that temperature plays a critical role in terms of providing a physiological signal and an objective signal per se in neuroscience applications, and it can be very valuable to evaluate and study in addition to other biomarkers, right? This one it provides a simple non-invasive measurement. It's the technology that is now available provides high resolution data, continuous, that avoids manipulating the animals, and also very scalable and can be integrated with other technologies to maximize biomarker evaluation. With that, I want to thank you for the opportunity, and I look forward to any questions either now or at the end of the presentation. Thank you very much.
Thank you so much, Jose.
Yes, thank you. So I will be talking about the Phenotyper, which is a versatile automated observation system that we have for behavioral phenotyping and compound screening within rodent models. The speakers for you today will be me. I'm Romain. I'm the Chief Product Strategist here at Noldus. I've been also within the account management and consultancy at Noldus, and I've worked at Hoffmann-La Roche in Switzerland. My background is in neuroscience and specifically drug development and neurohealth. On the other side, Lucas Noldus will be speaking at the end of this presentation. He's a founder and chief scientist, professor at the Donders Institute for Neuroscience at Radboud, fellow Netherlands Academy of Engineering. He has done his PhD in Wageningen University in Behavioral Ecology and his Master of Science in Leiden at the University of Biology, but I'm sure that many of you are very familiar with who Lucas is. So very briefly about Noldus as a company, we're a worldwide company focused on the development of software, hardware, integrated solutions. Founded in 1989 by Lucas Noldus. We have over 130 employees and over 14,000 customers in over 100 countries. And I would like to have a talk about the behavioral testing approach that we have. So we have conventional approaches to behavioral testing that often require different apparatus for each test. So the novelty response may obscure the behavioral effects that the animals will show. There's an impact of transportation and handling between home cage and test apparatus, which can impact the animal as well. And there can be many confounding factors on that. There's a big difference in how apparatuses are designed and how procedures are actually being done. So our alternative approach to that at Noldus is the behavioral testing in the home cage. So our idea is that we want to take the animal to the test apparatus. So we bring the test to the animal, and that's better in the reduction of the amount of stress. So we want to replace the battery of one-dimensional tests by one multi-dimensional test protocol. We want to investigate animals in a more natural environment, and we want to replace the human operator by an automated system. And lastly, very important, is testing in a dark period. And we want to observe rodents when they are awake, as that is where they are, of course, active. Brief introduction about the Phenotyper as it is. It's a home cage testing and hotel cage environment. There's zero human interference when the animal is inside. You can test single animals, you can test group housed animals. It is equipped with a video camera and infrared LEDs, and the camera is infrared sensitive, so it can still see under those conditions. It is 24/7 live continuous measurement of the animal's behavior. It is a rich and varied environment, and it's very suitable for optogenetics and operant testing as well. So then about the Phenotyper top unit itself, because it is so versatile. There's a modular tile system, and that allows for the integration of this camera module with IR, as I just mentioned. It can also have the adjustable white light. in there if you want. You can expand it with stimulus devices, which can be sensors, which can be feed-through devices. It has mechanical anchor points as well if you want to attach swivels, gantries, field body hardware. And it is compatible also with ultrasonic microphones, buzzers, speakers, and two-way communication modules. And all of that, of course, then integrates the integrated tracking and data visualization. That's, of course, the power behind it is that you are collecting data. So the output focuses itself largely on movement, behaviors, circadian activity, and consumables. So it is specific behaviors or patterns that an animal has over time. And I would like to present one scientific case study, and it's one of my, it's a piece of my own data that I've collected in the past when I was using the phenotyper before I joined Noldus, before I started working at Noldus. And this was about long-term whole range monitoring. And this was very suitable and very fitting actually to advancing the 3 Rs already through continuous observation. So we wanted to investigate how automated behavioral phenotyping in the catnap to knockout mice reveals stable phenotypes that could be missed by traditional short-term tests. So let's go ahead. So the challenge in behavioral phenotyping is that the traditional short-term tests that are available, behavioral tests, are often 5 to about 60 minutes, often shorter than 60 minutes. They may capture acute stress responses rather than stable behavioral phenotypes. So the requirements that we wanted to have while phenotyping the animals is that we wanted to have an objective measurement without a bias. We wanted to have an home-like environment. We wanted to have extended observation instead of short-term observation, and we wanted to have minimal handling of the animal throughout the experiment. So the case study model is a catnap to knockout mouse, which is a C56 black 6 background. It's an autism spectrum disorder model. Short-term findings are largely stable when it is about the excessive grooming, the rearing, and that they have a disturbed activity pattern. And the research question in this case was, do these persist in long-term observation? Well, we have the experimental setup, which is the phenotyper system, which was equipped with IR-sensitive cameras for 24/7 observation, an integrated shelter and running wheel. The shelter was infrared translucent, so that the camera and the light, the infrared light, would pass through it, and you could actually see the animal while being inside of the shelter. While for the animal itself, the environment would be dark and protected. Food and water was ad libitum, and it was a 12-12-hour dark light cycle. Protocol lasted more than 3 days. We had, because this was a proof of concept, 1 animal present per cage, but more animals per cage were the standard afterwards. We had 24/7 automated tracking. We had 2 experimental groups. We have a wild type and a knockout model. And the behavioral measures consisted of the distance moved, the amount of wheel rotations, the shelter visits, grooming, rearing, circadian patterns, and other data types that we've collected. So the first research, the first result was about the circadian activity patterns that we have. When we have the knockouts and we have the wild types, we have the knockouts with the blue line, we have the wild types with the black line over time. So the clock time in hour that you see below. The dotted environment at the left, that's the light phase, and at the right side, that's the dark phase, because we had a 12-12-hour cycle. And the key finding there is that knockout mice, they showed significantly higher activity during the dark phase in hours 22 and 5. So there were certain peaks of activity that they showed. But in the dark phase itself, hours 18 to 6, both groups show expected nocturnal activity increase. That's in general what we saw. But the knockout mice had some peaks. In the light phase, between 6 and 18, activity drops appropriately in both groups, indicating a preserved rest behavior. However, I did not include this here, but we did see that the amount of shelter visits during that time, so the amount of breaks during the sleep phase, were actually higher in the knockout mice as well, we found later. And the implication of this is that sleep patterns disturbances confirmed through continuous monitoring. And this is not observable in short-term tests because we're not observing the circadian activity. Then we had the running wheel, which was also present in the phenotyper, which automatically, when the animal is running inside of it, it measures the amount of rotations. It was very interesting. We found that knockout mice, they showed progressively increasing wheel running behavior over consecutive days. And the most striking to me was that in the first day, and actually also largely the second day, there was absolutely no difference in the behavior of these animals inside of the running wheel. But we saw that over time, so from day 1 to day 4, we saw a 2.8 higher use of the running wheel in the knockouts than we saw in the wild type animals at that point. So the wild type mice, they showed a very stable or slightly decreasing wheel running. It's a typical habituation response. But we did find in the knockout a very stable phenotype. So this stereotypic repetitive behavior phenotype would be missed entirely in traditional short-term behavioral tests. Now we have the circadian behavior distribution, and this was a very nice one for me to see as well, because we're very aware, of course, that the animal exerts most behavior during the dark phase. But by looking at the automatic behavior scoring that was done, more than 90% of all behaviors occurred during the dark phase. So it confirmed the proper nocturnal activity patterns in both the wild type and knockout mice. That's great. And the behavioral breakdown, we see that wheel running is largely done in the dark phase. Walking is largely done in the dark phase. But interestingly, at least to me in that point, was that grooming is a behavior that is actually done pretty spread up over the 24 hours. It even is grooming also while an animal is in a shelter. And it was good that we could see inside of the shelter as it was infratranslucent. So this grooming distribution is more evenly spread across the phases. And there was no excessive grooming visible in the knockout animals. That was actually a very stable phenotype in short-term tests. It was absolutely not a phenotype visible in the long-term testing. So the implication for this is that during the light phase testing, it's a common practice. It only captures 4 to 12% of the natural locomotor activity, and we're missing key phenotypes. And while measuring during that phase, we did find in the short-term testing some key differences, but they were absolutely not present in the long-term testing at that point. So when we compare that, what we had in short-term predictions versus long-term reality, so what we had in literature and what we found ourselves is that short-term, you would expect excessive grooming, excessive rearing, disturbed activity, and also some other unstable findings. But what we observed in the long-term, when we were home cage testing them for 3 or more days, we had no excessive grooming, and it was observed over an extended period. We had no excessive rearing observed over an extended period. We did confirm a disturbed sleep pattern with distinct activity peaks in the active phase and some more wakefulness in the light phase. And we had progressive wheel running increase over days, which was really interesting to find. An interpretation of this could be that short-term results likely reflect acute stress response to novel environments, even if you're handling and exposing and resting your animal for a long time before that, which I absolutely did. And that exploratory behavior normalizes once an animal acclimates. We found stable phenotypes, so it represents a genuine model characteristic, and repetitive stereotypical behavior is only visible in extended observation in this case. So our key conclusion at that point was that long-term measurements revealed stable and reproducible phenotypes that were less confounded by acute stress response, and that improved the translational validity, and that this reduces the need for repetitive testing as well. And at that point, I will hand over to my colleague Lucas.
Well, thanks, Romain, for the heads up, the introduction, and all the data presentation. I'm going to follow on to Romain's talk with a number of use cases of applications of the phenotyper in various studies relevant to 3R's concepts. One of the Achilles heels of modern behavioral science is reproducibility. The lack of reproducibility has often been mentioned as a weakness of behavioral studies. And we believe that standardization and harmonization of the context of study of the animal and the procedures and protocols contributes to better reproducibility of studies between laboratories and between treatments. Here we see the results of a study carried out between 2 locations, Aberdeen in the UK and Utrecht in the Netherlands, in which phenotypers were used for phenotyping of 2 inbred mouse strains, DBA2 and C57 Black 6. And what we see here is that the typical the activity changes during the dark phase and the light phase. The high activity during the dark phase, low activity during the light phase, were replicated nicely between Aberdeen and Utrecht. We see the same pattern here, with slight changes in activity between the strains, but overall a similar pattern between day and night. That also came back in the distance moved. We see here a clear difference between distance moved during first night and first light phase, the second light phase. Big differences between day and night, with an overall slightly higher activity of the DBA2, which replicated across Aberdeen and Utrecht. And we saw this in general changes between the Aberdeen and Utrecht, which were replicated nicely. So we're very happy with these results, to that say one phenotype performs similarly between two locations. A different question is if the effect of a compound can also be reproduced across locations. And this are results from a project we did with European labs and those in the US, with the partners in Groningen, Rijksuniversiteit Groningen, Pfizer in Boston, and Roche in Basel, in which a compound from Johnson & Johnson was used and applied to Shang2 knockout rats in studies on autism spectrum disorders, in which we found that the time spent walking, or the frequency of rearing, or the frequency of circling behavior differ between the wild types and the knockouts, but in a similar fashion across locations. So we saw, with increasing dosage, a decline of the time spent walking, but with the knockouts showing a higher say time spent walking than the wild types. We saw the same at Pfizer and same at Roche, and the same the decline across all these locations. And the same happened with the rearing and the circling. This is the first time that we were able to reproduce a compound effect across labs, a continent apart from each other, Netherlands, Switzerland, and the United States. A very different phenomenon is measurement of food and water consumption. That's also possible with the phenotyper by using the built-in feeder and water, the drink bottle, and it's where you can either measure the absolute consumption with the consumption meter monitor or the licometer attached to the drink bottle, or just as a proxy the time spent right in front of the feeder or the water drink, where we know that score is associated with the actual eating and drinking behavior. And here the effect of a compound can be with receptor antagonist AM251 was tested, which leads to a number of behavioral changes, not only in particular the time spent in front of the food zone, which we see here, the distance moved, time in front of the food zone, which was significantly lower with the treated animals compared to the control animals, and also food consumption went down. And it has also repeated itself night over night after repeated drug treatment. So lower Lambert walking activity and less time near the food zone, actually less food intake, which can be measured with the phenotyper over days. Well, so far we've talked about, say, spontaneous behavior, eating and drinking, but we can also use this phenotyper to the program, the automated delivery of stimuli, and measuring the response of animals to these stimuli. And for that, the EtherVision software comes with a built-in programming capability in which the conditions and actions can be played in a graphical user interface and connected with arrows, which lead to, say, automated scripts, whereby actions are performed by the system when certain behaviors or certain temporal or spatial conditions are met. So here we see the script of an experiment from start all the way to end. And when the certain condition occurs, then in the feeder zone, then an action is taken by the system, for instance, a trigger to the pellet dispenser to release a food reward, or with the drinking monitor, say a light can be switched on or sound can be switched on if a certain behavior occurs. this is just an example of a script when countless possibilities are possible. One example of the application of this capability is the anxiety testing through the light spot test, whereby animals are brought into a sort of a conflict situation by projecting a bright light spot in front of the feeder during the first period of the dark phase, when animals are normally becoming active and inclined to start eating. So here we see this dark phase, where starting at, in this case, 7:15 in the evening, when the light is switched on, and animals which are, say, the animals which are with whom this happens show a strongly suppressed time in front of the feeder, as opposed to animals who are not in this undergoing this protocol. So we see here 4 protocols: mice subjected to the light spot test, and this one is actually the one in whose case this happened, and in the other cases it does not happen. So showing that animals who are inclined to go to the feeder are actually staying away from the feeder because of this bright, frightening light. And this can be suppressed by compounds like valproic acid, which then suppresses this phenomenon, and leading to a sort of a rescue of this phenotype of the animal, showing its natural inclination to move to the feeder, even when the light is on. And another application, a very interesting one, is to automate the testing of learning in the phenotyper by using a very simple add-on, a piece of plastic called the cognition wall, a plastic triangle with 3 holes, which can be used to test discrimination learning and reversal learning in mice. And this has been developed and validated by a partner company called Silix, now part of InnoCer Laboratories, for testing the disease progression in Alzheimer models, the APP-PS1 mouse, using the fact that animals learn to take a particular entrance of the cognition wall, and be rewarded with a sweet sucrose pellet if they do so. So the protocol here is, if the mouse takes the left entrance and does that 5 times in sequence, it is rewarded with the sugar pellet. If it takes the right entrance or the middle entrance, that does not happen. Now, it takes a while before mice learn this, and a control mouse takes about 200 entries before it figures out that it has to enter the cognition wall that way. This can happen within a night. So the active mice are constantly taking these entrances, and after 200 entries, it makes 80% of the right choices. If this mouse is treated with MK801, which sort of induces a learning deficit, it requires about 400 entries. Now, this in itself is an interesting metric to assess or quantify learning capability. And this has been used in studies on Alzheimer, where we know from other methods that after 3 months of aging, the alpha-beta overexpression in the brain can be detected. The actual deposition in the brain takes about 9 to 12 months to be detected. That's also the time it takes before learning impairment in a water maze becomes manifest. But already after 3 months, the synaptic dysfunction is apparent. Now, if these mice are tested in the cognition wall, in the phenotype with the cognition wall, it turns out that the learning disability is already apparent in the APP-PS1 mouse after 12 weeks, and even stronger apparent after 16 weeks. So rather than waiting for 9 to 12 months before you can test your mice in the water maze, you can already find this phenomenon after 12 to 16 weeks in the phenotyper. So we're very happy with this result. And over the years, this partner company, Silix, has developed a number of paradigms for cognition and anxiety testing, as well as spontaneous behavior testing in the phenotyper, and published them all. So for all these diseases here, Alzheimer, you can read them at the bottom, Alzheimer, ADHD, ALS, ASD. These animal models have been developed through these paradigms and published in the literature to use this one and only, say, instrument for all these different paradigms and disease models. Now, after this system has been under development for 20 years, it was first released in 2004, and after 20 years, so about 20 years, we published a review of studies, and there are now hundreds of labs using this, and we're very happy with the take-up. But it takes a while. It takes two decades for this technology to become widely accepted. And wrapping up, we believe that this phenotyping system contributes to advancement of the 3 Rs through enhanced refinement. There is no more handling of the animals during the experiment. They can stay for weeks in the system without any human intervention. No transfer from home cage to novel arenas between the different testing of different brain functions. An enriched home environment with food, shelter, bedding, and drink. Reduction of animal testing by collecting more data per animal. So we need can obtain a higher statistical power with the same number of animals, and in the end, you need fewer animals to reach the same conclusion. And reproducibility is a big plus through automated scoring, standardized protocols, and standardized conditions, leading to stable allowing phenotypes to remain stable and to be quantified in an objective and standardized manner. This ends our presentation. Thank you very much for your attention, and I'd like to acknowledge the different projects that have provided funding to do the research that resulted in this system. Thank you very much.
Thank you very much, first of all, for the invitation to speak here. And secondly, thank you very much, Lucas, Romain, and Jose, which basically presented a lot of concepts behind digital biomarkers, a lot of advantages, which is truly also for our systems. And also thank you for the presentation of the RFID technology. So, because this is also a technology which we are using. So, as mentioned in the introduction of the webinar as such, there is a paper published a while ago, which you can find, in which you can find basically the definition of what is a digital biomarker, what is a translational digital biomarker. So the main take-home message is these are data which are generated automatically in the home cage environment of animals. And the animals are less disturbed or not disturbed at all, except of an RFID chip, which is injected into the animals, which guarantees you to identify the ID of the animal. And of course, we talked a lot about how translational these biomarkers are, and I have also a couple of examples here. So what is TSE Systems doing? So basically, we are long-term in the market and known for behavioral systems, mainly probably known the IntelliCage, which I will speak about as well, which is one of these fully automated home cage systems. Then we have a telemetry system, which is also capable to be used in group-housed animals, so that you have a telemetry system under group-housed conditions, which then measure ECG, EEG, EMG, and other biopotentials, including blood pressure and temperature. Last but not least, we have the PhenoMaster, which is a metabolic research system based on a classical home cage. And we are working on inhalation as well, which plays no role. But what we are really concentrating on is development of fully automated home cage phenotyping systems and the use of these kind of systems. So what is the vision behind this kind of home cage testing? So first of all, these are 24-hour test systems which observe your animal in a fully automated way. So that allows also that we reduce the impact of the scientist. So we reduce the fear, the anxiety which is induced by human handling, and also the human bias. And that leads basically to a reduction of data variability and an increased standardization between, but also within the lab. And this finally leads also to a higher animal welfare and saves a lot of money, animals, and power and costs. In the case of the IntelliCage, it is very important that the social component is back into the system. But let's concentrate first on the PhenoMaster, which is a system to measure indirect calorimetry. So we measure O2 and CO2 in order to calculate energy expenditure. And then we are trying to measure basically everything which has an impact on energy expenditure. So this is food intake, liquid intake, locomotor activity. We can put the whole system into a climate chamber, which allow you to do temperature challenge experiments and combine it with stellar telemetry. And when we talk about translation, then this is probably one example here, which is quite important. So this is a study from Eli Lilly, and it measures basically the effects of a triple agonist, glucagon, GIP, GLP-1, in mice, in monkeys, and in humans in the same paper. And what you can see is these classical effects of a reduction of body weight, a reduction of calorie intake, and also an increase in energy expenditure in a calorie intake matched control. So as I said, in the same paper, we have mice data, we have healthy human data, and show basically the same effect. And this is what we are calling translational value of the data. So we talked a lot about the limits of the reproduction of experiments, of data. And one of these reasons is basically the data variability. And it has been shown by Wilson and Krapp a long time ago in using an elevated plasmase that the human experimenter is one of the sources of data variability in behavioral data. But what else is important? I think it is also important to think about the social environment of these animals. So behaviors are seen probably only in dominant animals and not in subordinate animals. And there are also different physiological data between, or outcomes between subordinate and dominant animals. And therefore, it is very important to also include this social environment into the test situation. And one of the systems which is able to do that is the IntelliCage, which is fully automated home cage test system, which contains basically 4 of these operant conditioning corners. And each of these corners has an osmotic unit, has signaling lights. You have a door which can be open and closed in order to give access to the reward. You have also an air path for negative reinforcement. And as I said, you have 4 of these corners. And each of these, in front of each of these corners, you have an RFID antenna, which allow basically an animal to be detected. So, and these kinds of sensors and actors can be combined to a huge variety of behavioral paradigms, including all operant conditioning tasks, spatial learning tasks, etc., etc. And the animals can really live long time in that cage, and you can hold up to 16 animals, mice, in this case, in a single IntelliCage. So Lucas already talked about data variability. We show basically the same, that these are 2 different labs, 3 different strains, and you see that the data variability is reduced using the IntelliCage, and also the reproducibility is much, much higher than in conventional tests. And this leads basically to the same study which Lucas showed you. We have done using the IntelliCage in labs in Hamburg, in Rome, in Sweden, as well as in Switzerland. And as you can see, these 3 different strains look in terms of visits, in terms of reversal learning, pretty much similar. So there's a lot of questions in terms of long-term. Yes, we can measure long-term animals in that cage. These are examples of the measurement of locomotor activity, which you can measure by the number of visits in each of these corners. And as you can see here in this transgenic Alzheimer mice, you see that the regular things like habituation to the system, these are a dark period versus the light period. So the activity is higher in the dark period compared to the light period. The animals habituate to the cage, and you can also see this kind of classical age-dependent decline of locomotor activity if you compare this kind of plateau here in the 4-month-old animals compared to the 14-month-old animals. And you also have basically problems with the habituation in these old animals. So longitudinal studies have been published a lot using this kind of cages. Here, for instance, you see differences between young and old animals as well. In a place learning task, we train basically the animals to a single corner. See that the old animals are impaired in that task compared to the young animals, and they have also a problem with the reversal learning. So one of the big advantages of the IntelliCage is that you have a group of animals in that cage. So when we talk about anhedonia, which is a core symptom of depression, there are lots of people which basically measure anhedonia with free sucrose consumption in the home cage. But in order to do free sucrose consumption in the home cage, you have to isolate your animals, otherwise you don't know who's drinking how much. And if I put social isolation into PubMed, I would probably end up with 10,000 papers which will tell me that social isolation is a method to induce a depressive-like phenotype. In the IntelliCage, you have the big advantage that you can measure every single lick, and that has been done here. So you can look at the sucrose preference in that cage. And what you can see here in the control animal on the left side, that you have a nearly 100% sucrose preference in these animals. And when you add a chronic mild stress, then you have this drop down. So this IntelliCage can also replace certain classical methods. As you can see here in DICER1 gene inactivated animals, which show in the classical Morris water maze a memory enhancement. The same memory enhancement can be seen in a fully automated way in that place preference task in the IntelliCage. And not only memory enhancement, it has been also shown that in these Alzheimer mice, for instance, you have a drop in memory performance. One big advantage is you can also define fully automated test batteries. where animals go basically step by step without leaving the cage through a whole battery of different behavioral tasks. This is one example here of Ian McGregor's lab in Australia, where they perform, for instance, several adaptation states, and then a kind of a reversal learning, a continuous reversal task, which is similar what you do in the 8-arm radial arm maze task. If you want to know more about the IntelliCage, we have published a paper with a recent review paper with Hans-Peter Lipp, where you can find a lot of information about what type of research areas the IntelliCage have been used. So you see here, for instance, on the left side, it's aging, it's new developmental disorders, etc., etc. And there's also a list of all behavioral paradigms and a link to the paper which have been used this behavioral paradigm. There's also the IntelliCage is also made for rats a while ago. So the IntelliCage is, of course, also available for rats. It's just a bigger cage, and you can, depending on the size of the rats, hold up to 6 to 8 animals in there. And within that review, we also used this kind of corners for a study in marmosets. So that means that the same concept of behavioral setup can be used also for a variety of different species. And last but not least, so we also came up with a new system. Instead of having the cylinder here, you can also place the regular home cage in there. And this is an AI-based system in which you basically have 4 cameras which are observing the animals. And one of the big advantage here is basically the you do a 3D reconstruction of the animals. You have a variety of different body points, and based on these 4 cameras, you do a 3D reconstruction. And after this 3D reconstruction of the animal, the system automatically cluster basically in an unsupervised manner the behavior. And then you can basically watch this animal or watch these clusters and give them a certain name, which have been done here. And what you can see, this is in 1 hour experiment here after an acute stress. What you can see is basically that you have much more grooming in males and females after this first stress compared to the naive animals. So an unsupervised clustering, fully automated, observing these animals is possible with this behavioral atlas system. Finally, I thank you for the invitation and your attention. And if you have questions, you can go to our website or send me an email, whatever you prefer.
Thank you, Holger. So if I could have all of the speakers come back on camera, but leave yourself muted for now, we'll do the Q&A. So if you have any questions for all of the speakers or specific speakers, please put them in the Q&A box and we will answer them promptly. We do have one specific question for Roman. Can you explain how you did the statistical analysis on the 24-hour circadian locomotor activity?
Yeah, sure. Yeah, that's a perfect question, and it was expected to me as well. So I averaged the hourly distance moved across the recording days for each animal, and then ran a point-wise comparison between genotypes at each hour, and the asterisk indicates 0.05 p-value difference. I'm very aware that this doesn't correct for multiple comparisons. The goal here was more exploratory in identifying where in the circadian cycle the genotypes diverge, not putting direct conclusions of the specific differences in a specific hourly time point.
Great, thank you. Does any of these softwares track the behavior of 2 animals at the same time, as in the case of a novel social dyad test?
Sure, I could answer to go ahead. So EasyVision does indeed track multiple animals at the same time. We have a AI algorithm that automatically finds the differences in these two animals to actually give the animal a unique identification without any hardware identification with it. So it's like a visual marking, you could say, like a tail mark, for instance, or a small back mark. And in that case, indeed, the software will learn within 3 minutes what the difference is between those animals and make sure that all tracks and all behaviors are nicely separated between these two animals.
I guess I'll just add that with the UID mouse matrix, we're able to track group house animals, not particularly necessarily behavior. We'll do more locomotor activity, but temperature, we can track multiple, up to 5 animals in a cage. So depending on the resolution that you want, we'd recommend anywhere from 2 to 5. But just to be we don't do social behavior, although there could be ways that technology can be accommodated in different cages, more for a customized application.
Great. Thank you. Holger, did you want to answer that one before we move on to the next?
Yeah, we take, in terms of the software, we don't have a recording software on the IntelliCage. So this is basically based on the performance of the animals and the RFID chip in that corner.
Thanks. For those using computer vision analysis, how are you validating your vision platforms? Specifically, who and how are you generating training sets and how are you using them?
That's a answer I can answer very simply or very complicated. So let's try to find the middle ground here. So many of the sets of data that we have come from our users ourselves. So they have been made available to us for us to train specific models. These models are then also manually annotated, manually validated, so that we have a comparison between, so that we have a true data set, so that we have true behavior annotations, true body point annotations. Then the model is trained on that, and of course you expect it to work very well on the data it has been trained with. That is to be expected. But then we also validated along untrained data sets, so data sets that are not part of the entire training set, to see if the performance is up to where it should be. So then also these data sets are then manually annotated, manually labeled, and then compared there. And the accuracy of it depends on how far away it is from the environment in which it's trained. We see that many of our models, they are, to me, pretty surprisingly very generalizable, so that even if it's not specifically trained in a certain environment, that it still performs really well there. But everybody, of course, needs to be pretty careful with AI models to apply them in all types of environments that they are not trained for. But we do test it and we do advise on whether an environment is expected to be suitable, yes or no.
Yeah, I can add basically for our system, for the Behavioral Atlas system, that the user always have the control over the clustering as well. So that means that the whole video is basically segmented into the frames where you show this behavior or this type of cluster, and the user can basically look at this cluster then and define themselves if what type of behavior he see, or he keep cluster 17 as cluster 17.
Great. Thank you.
Yeah, so I'm really curious what sort of behavioral or physiological modalities people would be interested in seeing added to the systems we presented, the three of us. So for instance, ultrasonic vocalizations or heart rate measurement or optogenetic stimulation. And is anybody interested in, say, integrating that sort of technologies in their automated home cage monitoring system.
You can go ahead and reply in the chat to that.
So yeah, sure, and people can reply in the chat. Yeah, integration of whole body plethysmography to the phenotypers. Yeah, so that probably means photoplethysmography, whole body photoplethysmography for what, I'm curious, for what purpose? Because with photoplethysmography, you can assess, say, the blood flow changes of heart rate, changes in blood vessels by viewing skin color changes as a result of blood pulsation through the vessels. Say that is done in say in human face reading, where we can read the heart rate from someone's forehead skin. In theory, that can also be done with animals. I've never seen it done yet, but it's interesting. I'm curious if Mathieu Conroy is indeed pointing in that direction, because he says whole body plethysmography. I'm not so. It would be great to know what is meant with that. Assessing changes in respiration in animals. Okay. Well, that's a great suggestion. We need to look into that. Yeah. Thank you.
Great. Oh.
There's something pops in. May or may not be required. Well, that's a statement by Jordan Bruin that we can only happily support.
Yeah, for sure. That'd be great. Yeah, I agree. Very labor intensive.
Yeah, I think this comes with time indeed, that many of new test environments also that the different companies make and also academic institutes make. At first, there's not so much validation in certain outcome parameters, but we see over time that it takes 1 or 2 years and the validation comes in, and then these type of new tests, these new environments, they are validated and they do become the new standard. So it's probably indeed a matter of time.
Yeah, there's also this question in the chat here regarding the replacement of classical behavioral tasks in the home cage. Yes, it is possible. I gave you this one example of the water maze where you have this kind of comparison of probably a 10 days behavioral paradigm, which you have to run in the basement by running animals, let them swim for 1 minute, then they sit on the platform for 50 seconds, and you have to take them. It's very labor intensive. It's really time consuming. And of course, you can get basically the same results out of these place learning tasks from the IntelliCage. And that has been published several times in various reviews as well. So yeah, if you like, you can contact me. I can send you these reviews.
Great, thank you. So there was a comment about modifying the bottom of phenotypers to be easily removed for cleaning.
It's a good point. I worked with the phenotypes myself indeed, and it's a good point. They are really decent bottoms, but yeah, it could save you some time. Yeah.
And then we had a comment about scalability is very important. Also, we try to stick with the most well-validated simple tasks, locomotor activity, novelty detection over operant tasks. Certainly also more interested in physiology, respiration rate, internal body temperature are great.
I can address part of that. Yeah, scalability is it's actually something that we see as being very important. In our case, the difficulty is, I'm kind of going back to what Lucas proposed, in terms of what other parameters you want incorporated into the system, is that we find that there's complexity in the sense that every company has their own technology, that they're very, very reliable, and we focus on temperature. We try to incorporate video and other things, but we know that what we do best is temperature and RFID, so we kind of focus on that and let other technologies. That's why we seek more integration. I think that coming together different companies and integrating what they do best, I think that has a potential to being a better system, right? There's no need to replicate or, like I said, reinvent the wheel, right? So I think that kind of prolongs the development of products. So if coming together, I think that we see that as being more valuable. In terms of scalability, our system is very easy to scale up just because it's more simple, right? I mean, I think that a product like the IntelliCage, Noldus, and all that, it's a little bit harder because it's got more technology, more integration. So that's kind of my view. In our case, we can start with very few systems, and we can scale up to, complete racks in terms of if you just want to monitor very accurately temperature, and also incorporate locomotor activity. So we provide some basis of locomotor activity, not as complete perhaps as the other systems that are available, but then the advantage is that we do provide that scalability. So it really depends. I think that what we recommend is that kind of looking at what is the experimental design, what is the experimental goal, and based on those experimental goals, determining which technology would be more applicable. And in some cases, you may need to integrate different technologies. But so hopefully that answers the question.
Yeah, thank you.
Yeah, I can probably add to the IntelliCage that with one software license, you can run up to 8 IntelliCages, and if you have 16 animals in each cage, this is already a huge number of animals, which you usually do not run in a regular experiment. And on top of that, the system is very compact, so it's the size of a rat cage. So because limit in the animal facility is also, in terms of space limit in the animal facility, is always a topic.
All right, I'm not seeing any come up, so I think we'll go ahead and end the webinar. And thank you all for your talks. They were all wonderful, and we will get the recording out as soon as possible to everyone. And I hope everyone has a great rest of their day. Thank you all for joining.
Thank you.
Bye.
Thank you as well.
Thank you very much, and thank you for organizing this webinar.
Yes, of course.
Bye-bye.
Bye.
Bye-bye.